Today's presentation is about water quality and how we can look at the organisms that live in water to determine, to assess or determine how good the water quality is. So kind of obvious that if the water is so polluted or so poor, there wouldn't be anything living in there. So that's why just looking at what's living there is a good way to assess water quality. And scientists and ecologists have come up with a whole system of scoring and evaluating those organisms. So that's what the rest of this presentation is about. Just kind of this little graphic, I guess, shows how human activities alter the watershed. We've been talking a lot about watersheds. That's kind of the foundation of our whole aquatics discipline, is what happens in the watershed and how human activities influence water bodies within the watershed. And in this presentation, we'll be talking about how human influences on the watershed affect the animals that live there. All right, so one of the big ones that we use is aquatic macroinvertebrates. And that word can be, you can look at the different parts of the word to figure out what that means. And so aquatic means something lives in or near water. Macro, this is one that kind of messes students up a little bit. And when I ask students, what does macro mean? A lot of times they say you need to use a microscope. You can't see it. And actually macro means big. It means big enough so you don't need a microscope. So it's big enough to see with your naked eye. If it was micro, that would be like microorganisms. Think like bacteria and things that you need some kind of a magnification to look at. And at Prairie Waters, and when we're doing macroinvertebrate studies, sometimes we do use magnifying scopes, but we use those to magnify their body parts to make them easier to observe and identify. And then something that is an invertebrate is something that is without a backbone. So aquatic lives in or near the water. Macro is large enough to see with the naked eye. And invertebrate is something without a backbone. All right, and you can see I've put some examples up there. The top one is a crayfish. The second one down is a water boatman. The third one from the top is a dragonfly larva, and the bottom one is a snail. So yes, even though snails are also an aquatic macroinvertebrate, but it's not necessarily a bug. All right, so we call this looking at what lives in a water body. We call it a bioassessment. So again, we can break that word into parts. So bio means life, and assessment, you take assessments in school, and those are tests or evaluations that your teacher gives you to kind of just to see how you're doing. So a bioassessment is looking at what lives in the water to determine how that water body is doing for supporting life. All right, so some of the other points with that is it's looking at the diversity of animals that live in a water body, like a stream or a pond. I kind of alluded to this at the beginning of the presentation, that looking at what lives there, is there diversity? That's one of the best indicators of water quality. And also one of the first indicators, if you look at that and you see there's not good diversity of animals that live there, that's a good indicator that maybe we need to start running some more chemical tests and do a little bit deeper testing. And the second point, the middle one, is says each type of aquatic macroinvertebrate has specific conditions that it can survive and thrive. So dive a little bit deeper in on how we use aquatic macroinvertebrates. And then the third point, I put in a link, and I'm not going to show that because we've had trouble showing videos and going to external links, but I've given a list to Andrea, and you'll be able to watch that link. And that video is a really good one because it includes using fish for a bioassessment, using macroinvertebrates, and also it shows them collecting some water quality samples. So it's kind of the whole stream assessment. All right, so there are several reasons why we use aquatic macroinvertebrates for stream assessments. So one, they're affected by the physical, chemical, and biological conditions of the stream. So physical, those are things like, what are the stream banks like? Is the water flowing, or is it stagnant, slow moving water? Chemicals, think of, is there enough dissolved oxygen? Is there pollutants? What's the pH like? Those are chemical parameters. And then biological conditions. What kind of predators, or is there enough food for the macroinvertebrates? The second reason is they can't escape pollution very easily, especially when if they're fully aquatic or if they are a larva. They don't move a whole lot. So even if those larvae do eventually become adults that can fly away, they're stuck in the water, and they don't, of course, if you're a small bug, you don't walk too far away, unless you were carried away, I suppose, by currents. Third one, they show the cumulative impacts of pollution. So macroinvertebrates, even though they're macro, big enough to see with the naked eye, they're still small enough that it doesn't take a whole lot of pollution to impact populations, especially in those macroinvertebrates that are really sensitive to pollution. Fourth one, they show the impacts of habitat loss that might not otherwise be detected by chemical testing. Fifth one is they are an essential part of the stream's food web. So they are a source of food for other animals, and they also, some macroinvertebrates eat other macroinvertebrates. So they're all a part of the stream food web. The sixth one, some are really intolerant of pollution, and we're going to talk about that more. And even though sometimes we might struggle to identify them as novices, for the scientists, they're pretty easy. They're pretty easy to identify, and they're easy to sample. It doesn't take a lot of sophisticated equipment to sample macroinvertebrates. It's mostly just have a bucket with a screen in it and a D net and some waders. So not a lot of equipment to collect those macroinvertebrate samples. All right, so the next few slides are going to talk about pollution tolerance, and we're going to talk about which macroinvertebrates are intolerant to pollution, which ones are kind of tolerant to pollution or poor water quality, and then the last one, which ones are very tolerant to poor water quality. So on this slide, and I kind of misspoke when I say water pollution, because it's basically water quality conditions. So it could be pollution that's influencing that, but it could also be, is there enough oxygen for some of these macroinvertebrates, and is it an ideal temperature? Those are some examples. And the macroinvertebrates that are on your screen are the mayflies, stoneflies, and caddisflies. Those are all very sensitive to water conditions. So if these organisms are present in your stream or water body, they are indicators that the water is probably pretty good, because they wouldn't be there otherwise. And so just kind of to back up a little bit, when we are looking at macroinvertebrates, and even a lot of species in general, we use scientific names. And in the macroinvertebrate world, we refer to most of them down to their order names. So think kingdom, phylum, class, order. So the Ephemeroptera, those are the mayflies. The Plecoptera, those are the stoneflies, and the Trichoptera are the caddisflies. And if you take the first initial from each of those scientific orders, you get the EPT index. And that's again what I said earlier. That is looking at if you have mayflies, stoneflies, and caddisflies in your stream, that means there's probably good water quality. So there's a whole calculation index of looking at how the presence of these three orders of invertebrates. So we call that the EPT index. All right. Next up is the moderately tolerant invertebrates. And these are somewhat sensitive to water quality conditions. They don't need great water quality conditions, but they also can't survive in really poor water quality. So they're sensitive to pollution, and they need to have at least some a decent amount of dissolved oxygen and not temperature extremes. So they're found in streams with, like I said, fair to good water quality. And so the one to the left, that is the dragonflies, and dragonflies and damselflies both belong to the order Odonata. Dobson flies, I think another name for that is a fish fly. That is part of the order Megaloptera, and the Amphipoda are the scuds. And one thing, before I go much further, I do want to stress that we're not expecting you to know scientific names. When we give you a sample to identify macroinvertebrates, and I'll get to this in a second, there will be a chart that a key for you, a dichotomous key to key out and identify them, and the order will be written in that book. So don't worry about trying to memorize all these scientific orders. That's not necessary. I just put them on there so you can get kind of an idea, and we'll talk about that more in a couple slides. And then the last group, they're the tolerant macroinvertebrates, and they live in any kind of water condition. So they can withstand very poor water quality conditions, but they'll also live in really good water quality, in really clean water. So they're very, what do I want to say? They're very versatile, and they're kind of generalists. They can live anywhere. And so we've got the midges, the black flies, and the leeches. And I'm not going to even try to pronounce the order name for the leeches. I talked to my coworker today, and well, I had to write it down. So anyway, so these are very tolerant to poor water quality conditions. All right, so here's kind of a graphic, and these are all the different kinds of macroinvertebrates you might find in a healthy stream. And you can see that there's a lot of very sensitive macroinvertebrates. You've got some mayflies, that case-making caddisfly, and over here is a stonefly. And so you can see, though, that there is a lot of biodiversity. So there's lots of different kinds of bugs, not just the sensitive bugs, and there are a lot of total bugs. So high biodiversity and high abundance, lots of things living there, lots of different things living there, is indicators of a healthy stream. Polluted or poor water quality streams, you can see there's not much for biodiversity, only has those macroinvertebrates that are tolerant to poor water quality conditions, and there's not much at all abundance. There's very few and not much for biodiversity. All right, so we're going to move on to identification, and I'm going to go through some different ways to identify. We'll be using looking at a dichotomous key that I took. I found a PDF online that is the same dichotomous key that came out of this Streamkeepers Field Guide. And this field guide, again, you can contact Andrea, and she'll get a hold of Tina Harding, the other Aquatics member, and get you a free copy of the paper copy of the field guide. the Streamkeepers Field Guide. If you don't already have one, I strongly suggest you get your free copy. It's just a really good overall aquatics resource. And the Dichotomous Key is on pages 148 to 161 of that. And then this link with the www.bae, that's the online version of that Dichotomous Key from the Streamkeepers Field Guide. And again, Andrea will be sending out a document with all the links that I've posted in my presentation. And then this last one, it's macroinvertebrate pictures. I've just found it just looking online. It's kind of a pretty basic pictures with different macroinvertebrates and their order name. These, of course, these sources are in addition to the other biotics resources that we posted on the original aquatics resources that are posted on the Envirothon website. So these are just kind of some additional tidbits that you get just for watching this video. All right, so I'm not sure. Can you see, Tina, can you see this dichotomous key good enough?
I can.
Okay, you can. So this is that same dichotomous key that I was showing you a link of. And if you look, dichotomous keys are basically, I don't know if you ever read those books when you were a kid where you could read one chapter and it would say, oh, if you want to do this, go to this page. If you want to do something else, go to a different page. And that's what dichotomous keys are. They're just asking you questions about what this, in this case, a macroinvertebrate, what it looks like. And then that's going to lead you through a series of steps until you finally narrow it down to what your macroinvertebrate is. So I'm going to go back a couple slides and we're going to look at, we're going to look at this stonefly. So take a look at the stonefly, kind of take a look at what it looks like. It's got some long antennas. It's got legs, 1, 2, 3, 4, 5. It's got 6 legs. They kind of look like they bend, and a couple filaments that come off the back end. So now we're going to go back ahead to our key. And so step number 1, does it have segmented legs or not segmented legs? All right, so we looked, it has segmented legs. So it says go to step number 2. Does it have 6 legs or does it have more than 6 legs? All right, and we counted 6 legs. So that says go to step number 3. Did it have wings? Did it have wings or not fully developed wings? I did not see developed wings, so I'm going to go to step number 4. Look at the body. Was the body longer than it was wide? So yes, it's skinnier than it is fatter. Excuse me. So we're going to go to step number 5. Step number 5 says, does it have 2 or 3 distinct hair-like tails? Tails that aren't fleshy or hooked, but maybe fringed, or does it not? So yes, it does have 2 or 3 what looks to be like tails. So we're going to go to step number 6. Step number 6 says, does it have 2 or 3 tails that are plate-like or hair-like gills along the sides of the abdomen? If that were it, it would be a mayfly, and we'd have to go back and look. But if we go back and look, if it does not, if we go back and look, we can see that I don't see any kind of hair-like gills along the abdomen. If you look at this mayfly, though, you can see it does have those hair-like gills. So we'll go on ahead back to the key, and you can see that it only has 2 tails, and that it is a stonefly larva. And you can see if it did have those hair-like gills, it would be a mayfly larva. So those are 2 macroinvertebrates that are often confused when it comes to identification. And so you can see by going through the key and looking closely, we can identify those macroinvertebrates. And they were nice enough to also put sketch-like drawings. And you'll see Underneath the mayfly larva, it says it is the order Ephemeroptera, and under the stonefly, it gives the order of Plecoptera. So you can see when you look at those, you don't need to know those scientific orders. It'll be written in these keys. All right. And these are just the other 2 pages of the keys. You can look at it at your will. And again, this key is going to be linked in the links portion that I've sent to Andrea. So just kind of move through that. We can get an idea of water quality by scoring, by counting up what kinds of macroinvertebrates we found. So this is kind of a simplified table. There's a table similar to this in the Streamkeepers Field Guide. This one is a little bit simplified, and we wouldn't use anything much more complicated than this. So, so this would be, let's say you went out and you collected macroinvertebrates from your stream, and you brought them back to the lab, and now you want to see what kind, you've identified them, and now you want to see what are these macroinvertebrates telling me about my stream. So you look through, and you just want to put a check mark next to what you found. You're not counting how many. You just want to know whether they're there or not. So like whether you have 20 mayflies or 1 mayfly, you would still just put 1 check mark next to it. And on the next slide, I'll kind of show you a filled out one. So on this one, this particular sample said that there was, in the sensitive category, there were caddisflies and there were mayflies. So there were 2 check marks. And since they are sensitive and they're a better indicator of water quality, they are weighted a little bit higher. So you multiply the number of check marks times 3. So there was 2 check marks. I multiplied it by 3 and I got 6. In the medium sensitivity category, there were common net spinning caddisflies, damselflies, dragonflies, and scuds. So each of them got a check mark for being present, and there are 4 total. So I put there are 4 times 2, so they're weighted a little bit lower. That gives me 8. And then in the very tolerant category, there were leeches. So I only, that was the only one present. So there's 1 check mark, and 1 times 1 is 1. And so I add 6 plus 8 plus 1 is 15. And then at the bottom, you can give it a ranking. And so since 15 falls between 11 and 16, we can say that that stream water is fair water quality. All right. Now we're going to move on to fish. Fish can also be used as a biological indicator. But as you'll see when I get into it a little bit more, that there are some advantages and disadvantages to using fish. And all the information that I'm using is only from the resource, An Introduction to Freshwater Fishes as Biological Indicators. And that is the resource that we've posted on our aquatics page. And I put this resource again on today's link. But and that's a PDF. It's a whole book, but we've narrowed it down that you only need to study from pages 3 through 12. That's kind of where the most pertinent information is. All right, so here's a little bit of basic fish anatomy for you to know. And it's helpful when using the dichotomous key for identification, is understanding these terms for fish anatomy, like how is the mouth oriented? So on number 1 or A, it's an inferior, so that's like a sucker. The mouth is coming off from the bottom. So you can just you can look at these later on. I'm not going to go through all of these. You can see that there are different tail types. Again, those are caudal fins is another word for the tail. And again, you can look at the gills and the other body parts. And I've got a better diagram that I've put in here. This is just a little bit bigger and kind of, again, shows the fish parts. And we use those for identification. And I'll have to include, I don't think I included that link on my document. All right, so some of the advantages and disadvantages for using fish as biological indicators is that, I guess, as macroinvertebrates are small and they don't live very long, fish, on the opposite end, they live a lot longer. So the longer they live, the more opportunity they have to build up certain toxins or pollutants in their body. Also, fish occur in a wide variety of habitats, and they've been extensively studied on their habitats and their behavior. Also, there's a wide diversity of fish that live in different habitats, that feed on different certain organisms, how they reproduce, and again, their tolerances to different environmental stressors. Again, these are easy to identify, relatively easy to identify compared to some other aquatic biota. So one thing that we're not using for our aquatics is we don't require you to look at any algae or anything. That's you can use algae as a biological indicator. We don't do that because algae are really complicated to look at and to identify. And so going on, so those are the advantages to using fish, but part of their disadvantage is it takes a lot of manpower and sampling equipment. You can either use electroshocking, where somebody goes along with this little electrical probe and it gives a mild electrical impulse that kind of stuns the fish enough for the sampling technician to pick it up and put it in a bucket for identification. But yeah, that's kind of some sophisticated and expensive equipment, and it takes a lot of manpower. And if you were to do just seining with a net, again, that's pretty hard, especially if the water body has a swift current. And then the other one is migratory. So fish can move a long ways. They have the ability to escape pollution or poor water quality in a certain area. They have the ability to migrate and move out of that area. Also, some fish migrate for their life cycles and reproduction. So just fish are a little bit trickier, and they have some disadvantages to using them for biological indicators. All right, and in that resource, just some terms to know. Biological indicator, I've talked a lot about that. So these organisms, or fish in this instance, they're derived from measurements and kind of like, and has no statistical properties. So if you look back at that diagram that I used back here for the macroinvertebrates, there's something like that for fish. Just kind of know that it exists and what it means to be a biological indicator. You don't really need to know much more than that. What is the biological integrity? Like, is this habitat capable of supporting and maintaining a community of organisms? So, and then an indicator organism. So that's an organism whose characteristics point out or directly point to environmental conditions. So I don't know if you've ever heard, but like a canary in a coal mine. So back in the old mining days, if they weren't sure if the mine was safe to go in, they'd put a canary in, and if the canary died, it meant that was an indicator that it wasn't a safe place for humans. And if the canary lived, it meant that it was probably an okay place to be. So that's what an indicator organism is. And ecological health is a system of assessments where they can determine, again, if the conditions are suitable to support life, and if the habitat is capable of mending itself and supporting a community of organisms. And lastly, when we're looking at fish, they are part of the food web also. And many fish are predators. They have lots of different feeding types, as do macroinvertebrates. I didn't go into the feeding groups of aquatic macroinvertebrates. That's something you might want to look at a later date. But for for fish, there are piscivores, which are fish that eat other fish. Herbivores, I'm sure you've heard of that. Those are fish or animals that feed on plant material. Omnivores can feed on anything. They can feed on animals or plants. Insectivores only eat insects, and filter feeders, they filter out the plankton and use that for food. Invertivores only feed on invertebrates, so they can feed on insects and mollusks or crustaceans. And then there are generalists that are known to feed on all fish and macroinvertebrates. So that's just kind of some terms you can look over and kind of put that together with our various fish. All right, so I was amazingly able to keep within my 30-minute presentation time. Are there any questions? And again, this will be posted online, and you can always contact us with questions about the presentation. Oh,
that was well done, Bonita. Thank you. And let me just remind everybody one more time that these are posted on the Envirothon website. You can download them and watch them as many times as you want. Again, let's reiterate that if there's any published materials that you would like to have, please don't hesitate to reach out to Andrea or Bonita or myself, and we will make sure that your advisor gets that material for you.